Hello, my name is Vince Davis. I'm the Annual Cropping Systems Weed Science Extension Specialist in the Department of Agronomy at the University of Wisconsin-Madison, and I want to talk about herbicide, mode of action, site of action, and a little bit of troubleshooting so we can understand the symptomology that herbicides cause in crops and why and how they're different. So let's get into this today, and again, we're going to talk about herbicides, how they work, and the injury symptoms they cause. So we're going to talk about that. Before I get going too far into that, I want to make sure you're aware of three other resources that I think are really important to have when we're talking about herbicides. The first is the Crop Data Management Services, or CDMS. And that CDMS website, you can search and get the herbicide label for, or the label for every herbicide available out there, and you need to always read and follow the label directions of herbicides. So that's the most important thing to herbicide use, is to read and follow the label directions. Now, in relationship to how herbicides work, a great website out there that explains in an interactive fashion to learn herbicide modes of action is the Purdue Weed Science website. There's an interactive tool they have that shows you the different herbicide modes of action, and then shows you symptomologies and how those herbicides work. So I think that is a great tool. And the last source that's important for you to study for CCA or anything else is the Weed Science Society of America, which houses several useful educational pieces on herbicide resistance, as well as all of the official names for herbicides and herbicide classification. So there's a number of useful resources you can find at the WSSA website. So I wanted you to be aware of those. Let's jump into herbicides though, and let's first off define mode of action and site of action, because there's a subtle difference, and weed scientists often use these interchangeably, but they do mean a little bit different things. The mode of action describes how the herbicide controls a plant. So it's how the herbicide controls a plant. It's a little bit larger category than the site of action, because the site of action describes specifically where the herbicide acts within the plant. So there are sites of action may be different within the same mode of action. So mode of action is a more broad category than site of action. Both have different specific meanings. We're going to get into that as we go forward. There used to be, not all that old, you can probably still get to this on online resources. We had the corn and soybean herbicide chart, and as an example here, you'll see modes of action listed in the furthest left-hand column, where the sites of action are listed within that mode. And another important thing to understand is the site of action is also can be referred to as the group number. Now, the group number is a number that it was generated by the Herbicide Resistance Action Committee, or HRAC for short, and that number was designed to make a much easier system for growers and practitioners to use to know that they are rotating sites of action in their crop management system, so they can just remember that they're using a group 1 and a group 9 versus trying to remember the longer names that are associated with that site of action, like ACCase inhibitor or ALS inhibitor, which may be confusing to some people that don't know what those terminologies mean. Now, why would you want to understand mode of action? And again, I may use mode of action and site of action interchangeably here, but why would you want to understand mode of action or site of action? There's several reasons. One is to plan weed management programs. Obviously, matching the right type of herbicide to the right type of weed problem, whether you're dealing with an annual or a perennial, could be very different in management systems. Also, you want to understand the mode of action to correctly diagnose crop injury symptoms in the field. That's probably, for this purposes, the most important reason to understand that mode of action. We're going to talk about how these herbicides work and how to identify their injury symptoms. He also helps to understand mode of action to avoid or understand potential interactions with other herbicides, such as antagonism or synergism. I'm probably not going to talk about that much in this training segment. I'm going to focus more on how they work and specific diagnosis in the field. There is, I believe you got a URL for those of you training for the CCA to get to this herbicide mode of action key, but this is available online, and this is a key that is produced to help identify when you are trying to figure out what went wrong with a herbicide in a crop growing situation, where that damage is coming from, whether it's coming from a herbicide and what herbicide. And so there's these flow charts out there that help with this, but we're going to talk about some of this in more specifics, but there are tools out here that you can get to help with that. When I talk about herbicide mode of action and site of action and link that to the damages they cause in crops and their mode of action, I like to make sure I go over this one important question, and that is, what is the number one most useful aspect of herbicides? And in my opinion, the most useful aspect of herbicides is selectivity. A lot of people may instantly answer the most useful aspect of herbicides is they kill weeds, but actually we can kill weeds many different ways. Tillage is a very effective way to kill weeds. You can kill weeds with flames or heat, freezing. There's a lot of different ways to kill weeds, but herbicides give us the ability to kill the plant we don't want, or the weed, within very close proximity to the crop plant that we do want. And so that is a selective nature of those herbicides. So we're always trying to achieve selectivity. That is, we're always trying to achieve weed control without damage to our crops. But obviously, understanding how those herbicides work is important, because when we lose selectivity is when we often get damage to crops. The second most useful aspect of herbicides is residual control. We can't get residual control out of tillage unless we do repeated tillage. So the ability to use a herbicide and then get control for an extended period of time is really important, because we want to get control all the way through the critical weed-free period, which I discussed in the last training video. But we want to protect the crop from weeds for an extended amount of time, and we get that from residual control. Because selectivity is so important with herbicides, it is the reason we have so many different herbicide classifications and so many different types of herbicides, because they all do different things and are safe or harmful to different types of crops, and are safe or harmful to different types of plant species. So understanding all that can be a lot to undertake or have to do, but because we're always trying to achieve that selectivity is the reason we have such great diversity in our herbicides that we use. So we have herbicides that we classify as primarily post, or they may be flexible in terms they can be used post or soil applied, or some herbicides are only effective when they are soil applied. We have herbicides that are translocated via phloem or translocated only via xylem. We have herbicides that only control broadleaves, herbicides that only control grasses. So we have different levels of classification. We'll talk about some of these. Because we have so many different herbicides, that's why we spend so much time talking about herbicide classification. I already showed you the corn and soybean herbicide chart, which was a predecessor chart to this chart here, which is available at the takeactiononweeds.com website. And this again shows the mode of action in the most broadest categories, but breaks down different sites of action of herbicides, and within those sites of action, the chemical family and the specific active ingredients in many different herbicides. And on the right hand side of this chart, it also shows the number of different herbicide sites of action that are included in popular herbicide premixes. So this chart is available at takeactiononweeds.com. We'll talk more about that later. But to go through some of these real quickly, the group 9 herbicide is the most common. Group 9 is glyphosate. It's the only herbicide in this category, and it is an EP/SPS synthase inhibitor. So group 9 herbicides share the same mode of action as group 2 herbicides, in that we say both group 9 and group 2 herbicides are amino acid synthesis inhibitors. That is their mode of action, or how they work. They work by inhibiting important amino acids, but they work at two different sites of action, where the group 2 herbicides inhibit the production in the ALS enzyme, or acetolactate synthase enzyme, where glyphosate inhibits production in the EPSPS synthase enzyme. So because they attack at two different locations in the plant, they're different sites of action, even though they kill the plant in the same way by inhibition of an amino acid. The group 1 or the ACCase inhibitors is a good example of herbicides that are classified as grass herbicides. So these herbicides are only effective on grasses because of the way they work. They are completely ineffective on broadleaf herbicides, where glyphosate, as I just discussed, is classified as a non-selective herbicide. It's actually effective on everything to some degree, unless it's been genetically modified, like in the case of Roundup Ready crops. I've already spent a lot of time just talking about the group 2 or the ALS inhibitors. Again, classified as ALS inhibitors because it inhibits the production of essential amino acids in the ALS enzymes. This is our largest group of herbicides. It is also our most diverse group of herbicides in terms of selectivity. It's also, unfortunately, the class that has the most cases of herbicide resistance in weeds. So it's a very large and important class of herbicides to understand, both from a use as well as a resistance standpoint. Some more different classes we have would be the growth regulators, or the group 4 herbicides. These would be herbicides like dicamba, 2,4-D, MCPA, very important classification of herbicides. We call these the synthetic auxins. Below that is our photosynthesis inhibitors, or group 5. We have photosystem 2 inhibitors, which include important herbicides like atrazine and simazine. However, we also have photosystem 2 inhibitors that are site of action or herbicide group 6, because they work in a little bit different site, but important herbicides like Basagran and Buctril. And more photosystem 2 inhibitors, but binding in a different location, are the group 7 herbicides like diuron or linuron, which are more amide and urea family compounds. Group 10, we have glutamine synthesis inhibitor, or glufosinate, also known as Ignite or Liberty. We have a classification of pigment inhibitors, which includes two different groups, or site of action groups, 13 and 27. The 13 is clomazone or command, important compound in broadleaf crops like pumpkin, also rice, and in some areas soybean, as well as HPPD inhibitors. Also, this is our newest classification, or newest mode of action, if you will. And important herbicides in there like mesotrione and timbotrione and toporamazone. So very important herbicides there. We also have cell membrane disruptors, and within the cell membrane disruptors we have sites of action 14 and 22. That includes important herbicides like famecifin and lactofen and aceflorofin, as well as the photosystem 1 herbicides in site 22, which would include non-selective compounds like paraquat and diquat. We have seedling root growth inhibitors in group 3, herbicides like sonalan or pendimethalin, prowl, trifluralin. These are herbicides that act by inhibiting root growth, where in contrast to that, we have soil applied herbicides that target to inhibit shoot growth, several sites of action here, like the 8s, 15s, and 16, 8, 15, and 16 different compounds in here that are broadly used, like acetochlor, alachlor, metolachlor, dimethenamide. So this group 15 is probably the most popular among a lot of these in corn, soybean crops, particularly here in Wisconsin. So you can refer to that chart, and it helps break down all of the different things. But going back to the reason we have all of those different herbicides is because we want selectivity. We're always striving for selectivity. A good example of the importance of that selectivity is glyphosate. I already made mention of Roundup Ready crops, and Roundup Ready crops have become the most widely adopted technology since the mid-90s when Roundup Ready soybeans come on the market. And the reason it's so widely adopted is because Roundup or glyphosate was very effective because it was non-selective on a wide range of weeds, but is not harmful to the crops because of the genetic engineering. So we engineered the selectivity into that cropping system. But for many other systems, we're relying on that important selectivity through a wide variety of mechanisms. We'll talk about some of those. And whenever we lose that selectivity, we risk having crop injury that we have to understand where that crop injury is coming from. So we're always trying to achieve selectivity with our herbicide use. We may achieve that selectivity by the placement or differential placement. Sometimes that comes from rooting depth. So many statements on herbicide labels and related to particularly the soil applied herbicides in terms of how deep the seed should be planted underneath the herbicide placement. Again, because we're trying to get separation of that herbicide to seed interception to avoid early season crop injury. In other cases, like in the case of the ACCase inhibitors, which only control grasses, we achieve differential susceptibility based on how the herbicide works. So because the site of action is distinctly different between monocots and dicots in this particular pathway, then the ACCase inhibitors are basically completely ineffective on broadleaf species. So we can have differential selectivity based on the site of action in which it controls. One of the most popular, or one of the most common ways of trying to achieve selectivity, though, is through the rate of metabolism. And what I mean by that is the herbicide still has can still have some effect to the crop plant, but the crop plant has an ability to metabolize that herbicide and deal with that intrusion of the herbicide in the system. But if the crop plant can't metabolize that herbicide fast enough, it may still encounter negative effects from that herbicide. One of the most common scenarios that we see this is with the grass shoot inhibitor or grass herbicides, or the seedling shoot inhibitor herbicides when used in corn in cold, wet soils early in the spring. If the corn plant is not able to grow fast enough or have enough rapid growth rate, you'll often see increased herbicide injury because it's not able to metabolize that compound quick enough. If we get into some examples of crop injury here, just to show you the importance of the specifics of how these herbicides work when you're trying to diagnose crop injury. On the image here, what I want you to take note is some very important things to keep track of or to assess when you're trying to assess damage is whether or not the newer growth or the older growth of the plants is most affected. In this picture I'm showing you here, you see the newer growth of the soybean leaf actually looks just fine, with the exception of some insect feeding that we're going to ignore right now, but the newer growth looks fine, where the older growth is actually the leaf that is most affected. This here is an example of a herbicide that was translocated via the xylem, and so it's affecting the older growth, but it's not translocated via phloem, which moves food from a source to the sink, or moves the sugar in the plant to the most actively growing regions of the plant, which would be this trifoliate here. So because the herbicide is moved in the xylem, it's affecting the outer edges first, and it's also affecting the inner edges, leaving the margins that actually are moving the material less affected. So you see the veins look okay, with the regions between the veins being the most affected and the outer tips working back in. This is a very classic herbicide injury symptomology from a triazine or a PS2 inhibitor herbicide. This would mimic atrazine carryover or metribuzin damage from a late spray application to soybean here. Another thing I want to point out is that the distance between the necrotic lesions and the green lesions tend to be right next to each other, and that's going to be in contrast to some other damage I'm going to show in a little bit. This is just a little bit more of the same type of damage, showing the outer leaves affected first, and then the inner veinal regions being most affected by necrotic lesions. Again, this is actually damage from a metribuzin, an inappropriate metribuzin application on soybean. A little bit different symptomology to look at here is soil applied damage from a PPO inhibiting herbicide. The PPO inhibiting herbicide has caused the inability of proper leaf formation on all the leaves, and so it causes this puckering symptomology here that you see, but again is affecting the older leaves more than the newer growth. So it's another example that I wanted to show where the newer growth is just fine, and this particular plant is probably going to outgrow this damage without much difficulty, but this soil applied PPO is affecting the expansion of all growth in that leaf, and because it affects the expansion of all growth, it causes this puckering of the entire leaf area. That is going to be in contrast with some other symptomology I'm going to show you soon. Now here is an example in contrast to those last couple examples. Here's an example of damage from a herbicide that's actually affecting the newer growth or the growing points of the plant. So here now, if you see the older leaves down here look fine or look better in this picture, but the newer growth areas are actually affected. This is a classic example of an ALS inhibitor, or herbicide with the mode of action that inhibits an essential amino acid. These herbicides tend to take longer to work, both ALS inhibitors and EPSPS synthase inhibitors. They take longer to work because the plant takes longer to feel the effects of an essential amino acid missing in the newer growth areas of the plant. So it affects these newer growth areas of the plant. You see this yellowing, absence of the whole growth. And with the case of ALS inhibitors to broadleaf crops, if you flip a leaf over, you actually see a very classic symptomology here of these veins being reddened and being very dark, which is very abnormal. So this is very classic injury of an ALS inhibitor because it's lacking essential amino acids that it needs to sustain continued growth. Now, another very popular herbicide symptomology is a symptomology you get from growth regulator herbicides. Here's an image that shows growth regulator herbicides, and from growth regulator herbicides like 2,4-D and dicamba, what you often get is what we call epinasty, and that's kind of an uncontrolled twisting growth of the above ground plant. And then you also get this restriction of growth called cupping. And so you see these leaves here are cupped, or actually form cups, because the whole leaf is puckered and the outer edges curl up. With dicamba, these outer edges often curl up, where with 2,4-D, oftentimes it will cause the leaves to malform and they will curl down. With growth regulator herbicides, you also tend to get this strapping. If you look at this leaf here, I'm trying to outline it. You get leaves that tend to be more linear in nature than they otherwise would be. Another thing I want to point out is that this is another really good example with the growth regulators where they tend to affect the newer growth of the plant, again because they're translocated via the phloem, not the xylem. It is the newer growth of the plant that is most affected, where the older growth down here in the plants are actually less affected. This last image here is a classic image from a seedling growth inhibitor, where you can see an absence of growth in the main vein here as you go out the leaflet on the soybean plant, and it forms this heart-shaped leaf because the growth in these outer edges did not cease, but the growth here where the reproduction of cells was stopped by the mechanism of action of a seedling growth inhibitor. And we often call this a drawstring effect, a drawstring effect or heart-shape effect of these leaves. Again, occurring more on older plants or older leaves than the newer leaves, but very classic to the chloroacetamide herbicides like dimethenamide, metolachlor or acetochlor, alachlor, those types of herbicides. Now, we also have to assess the way contact herbicides work in plants. So contact herbicides work, or post-applied herbicides work by coming in contact with the plant, but then also being absorbed or moved in the plant. So contact herbicides often destroy cell membranes. Here's an example of a PPO inhibitor, and what I want to point out are these red ring lesions. This is pretty characteristic of the PPO inhibitors as well as the PS1 inhibitors, because what they do is they work by destroying the cell membrane and causing toxic internal cell substances to leak out and then destroy the cell membranes around them until the concentration of herbicide inside those cells no longer cascade. When the concentration is no longer great enough to cascade and kill the next cells out there, and so it leaves us very characteristic red ring lesions. And again, this is characteristic of contact herbicides, but also shows the importance of spray coverage to effectively use contact herbicides. For contact herbicides to kill the entire plant, there has to be enough coverage to get all of these necrotic lesions connected to each other to actually kill the plant. Plants like this will eventually probably grow out of it in the new growth, because again, this will only affect the growth that is sprayed on or older growth. It won't translocate to newer growth herbicides. This was actually a diagnostic training with sharpen herbicide creating these red ring lesions, which was not a labeled application over the top of herbicides. So just done this here to show you how it works and the symptomology it causes. The reason I wanted to go through that is because when you're actually in the field troubleshooting some of these things, the interaction between management and herbicide placement can be very important, and this is just one good illustration of that. The lesions on this soybean hypocotyl here actually is from a PPO herbicide, showing these red ring lesions and the damage where it came in contact with the herbicide. However, this was done by a soil applied herbicide following all appropriate label directions, but what happened in this particular scenario is a herbicide was placed on dry soil, and the soybean was planted at the recommended depth, and there was no rainfall received, so the developing seedling was able to emerge, and as it was emerging, the first rainfall fell and actually splashed soil solution up on this developing hypocotyl. So you actually got herbicide damage from a rain event afterwards. So it's kind of an interesting showing that in this case we were trying to achieve selectivity or separation from crop to herbicide by placement and timing of that herbicide, and it didn't work in this particular scenario just because of the weather effects from this situation. Another example I often show to show that you can solve troubleshooting problems by understanding selectivity. This here is an example of a Liberty application or a glufosinate application on a Liberty Link crop. However, there was seed contamination where some of these plants did not contain the Liberty Link gene to make them tolerant of that application. So if you can understand why we lost selectivity here, and in this case we lost selectivity because of genetic differences, because of seed contamination, you can solve troubleshooting scenarios much quicker if you understand how did we lose that selectivity, if you will. So that's the end of the presentation for understanding herbicides and the fact that we have a lot of different herbicide mode of actions you understand and different ways they work, which will interact with how they're used properly to avoid crop damage situations.